showcasing gardens and nature in the valley and beyond. This is Let's Grow There, a WKBN Plus exclusive. Let's dig in. Hello and welcome back to Let's Grow There. I'm your host, Laurel Stone, and today we're going to be discussing something that is the backbone of crop production, but you might not know about what happens behind the scenes. My guest today is Nicholas Kellar, who has a degree in horticulture and works in this industry. Nick, tell people a little bit about what it is that you do.
Hi. So one thing that I do is tissue culture, it's a laboratory technique. We use this technique to grow and maintain cells, tissues, small pieces of organisms in a controlled, sterile environment outside of the original specimen. It involves placing a small piece of tissue onto a nutrient-rich, hormone-rich medium and maintaining it under a very controlled environment.
Now, Nick, you reached out to me to be on this show. Why did you do that? Why do you think people are interested?
Tissue culture is something that is becoming a very emergent industry. It's in the backbone of many food crops that we use, many ornamental crops that we use. So it's just one thing that I want to get out there more, talk to people about. It's something that when I was coming upon it was almost a taboo subject. It wasn't really talked about. There was not many resources to learn about this process, and I really love just sharing it. It's something I'm very passionate about.
Why would it be taboo?
It's usually confused with GMO, genetically modified organisms. One thing I would really like to point out is this is a very different process. There's no modification done to the plants. We use the hormones that are naturally within the plants, nutrients similar to fertilizer that you put on your lawn or your ornamental crops.
Now, explain to me in the simplest terms what tissue culture is, what you do.
So, like I said, tissue culture is really just taking small explants. In a laboratory environment, we call the organism an explant. So we take those small explants, we excise them down to the specific organ we're looking for, whether that could be a meristem, a node, a leaf culture. There's many options where you could use this. We take those organisms and grow them out.
For what purpose?
There's many different purposes. The main purpose is for pathogen remedy. So if a pathogen is present with any plant tissue, because we take such small pieces, usually they are not connected to the vascular system of the plant. So the vascular system is what these pathogens like to hijack and use to spread throughout the plant. So since we're taking such small pieces, usually the growth point of the plant or the apical meristem or apical dome, these pathogens are not present from that tissue. And then we can regenerate these tissues through something called totipotency, which is that a single cell can give rise to an entire plant.
Now, you're working on a really, really small level here. What is the big picture result of what you're doing? What problem are you looking to fix?
We are honestly trying to get rid of just the pathogens that are present. These pathogens can really destroy a crop. They can destroy plantations. If you get Fusarium wilt, for example, in your field, within a year, you can take out that entire crop. Fusarium takes upwards of 20 years to remedy out of a field, so your field is essentially useless at that point. With food scarcity being on the rise and we're losing viable farmland on a regular basis, this process can really help curb that.
Now, is this something, do you work directly with farmers who have problems, or are you working proactively to fix things before it becomes more of a problem?
Both. So proactively to produce clean stock that can then be seeded out and used for these crops, or for hard-to-find crops or specialized plants that may not be able to grow everywhere. Ghost orchid comes to mind. That's something that we could just take, remedy, and grow it out.
Tell me a little bit about the history, how something this technical got started, how it evolved.
So originally the theory of totipotency was coined by Gottlieb Haberlandt. I'm sure I messed up that name. He was a German physicist, but he one day was like, I wonder if I can grow an entire plant from these cells. And then in 1907, we had the first successfully cultured animal cells. And then we kind of didn't do too much for a while there. This was very practical. A lot of people weren't doing it until the 50s, where you had a fellow named Mr. Scooge and Mr. Miller who discovered that auxin to cytokinin, these are hormones that are found naturally within the plant. They tell the plant what to do, similar to estrogen or testosterone in humans. They discovered that the interactions dictate what happens to the plant. So then after the 50s, it became rapid advancement. In 1951, HEPA cells were formed from Henrietta lacks, which is pretty much the basis of most vaccines, and a lot of research we have into the human anatomy was formed. And then the 60s onward, we started creating full plant regeneration, and now it's just everywhere.
Now, is this something that people can do in their home, or this is really, your environment is sterile, it's a lab? Like, tell me what it requires to do something like this, because really you're in the scientific field.
Yeah, so in my lab, we use a laminar flow hood. So laminar flow is a laminar flow of air, which means the air is undisturbed. This is pushed through a HEPA filter, so this filter sterilizes about 99.99% of contaminants out. So there's always some variable in there. There's always a risk of contamination from a yeast or a bacteria or a fungi. So since this is so sterile and the medium that we use is very nutrient-rich as well as sugar-rich, a fungi or a bacteria could rapidly take over. That being said, I honestly learned to do most of this process at home in something called a still air box. So as long as you have air not disturbing where you're working, a baby food jar, and some nutrients, you can tissue culture at home if you would like.
Now, is this something, I have various house plants, some of them do better than others. So if I have a bad house plant, am I somehow able to use tissue culture and diagnose what's going wrong with that, or even something out in my garden?
Definitely. I'm pretty sure chances are if you bought an ornamental, Costa Farms comes to mind. You know, all their plants are also tissue cultured. So chances are it's already been through the tissue culture process. And a lot the procedures that we use are called protocols. A lot of these protocols are widely available on the internet. You would just find whatever plant specifically you're working with, look up its protocol, and you can find the hormones that you need to use, the nutrient regimen you need to use, and the grow environment that it likes.
How do you get into something like this?
Well, you have to be a very big plant nerd to start. I'm a lover of anything that grows, and the industry that I was working in was struggling with a very viral disease, and it was just something that I had already done at home, working with ornamental plants. So I kind of got pushed into a lab environment to work with a high-value crop, and we remedied that high-value crop, and I just fell in love with the process. I love to remedy these plants and see them healthier on the other end.
That's really cool. Thank you, Nick. We're going to take a little quick break, and then we come back. We're going to talk even more about the technical aspects and what his job looks like from the day to day. We'll be right back. Hello, and we are back. I'm your host, Laurel Stone, joined by Nick Kellar. Now, Nick works in the tissue culture industry, and I'm going to let him tell you a little bit about what his day-to-day looks like in his specific job. Nick, go ahead.
I work in the tissue culture industry. I work for one of the largest producers of cannabis in the state. Part of my job on a day-to-day basis is selecting a donor plant, which is just a mother plant or a stock plant that we want to initiate. So we will take small cuttings from this plant, usually nodulal segments and meristems. So your nodulal segments are anything that comes from a leaf to a branch point, and your apical meristem is anything that is a growth point. So you have shoot apical meristems and root apical meristems. I mainly work with shoot apical meristems because they are the most vigorous and they're the easiest to work with. So we'll take these explants. Again, an explant is just whatever plant you're working with in a laboratory environment. We wash them in a soap solution, usually for about 20 minutes, but it depends on the crop that you are using. This is just to get any debris that may be present on the plant tissue off. After this, we wash them in a bleach solution, 10% bleach. Some people use ethanol. You can use silver nitrate. Bleach is the most common thing that you're going to have at home. This is going to kill any pathogens that may be present. Since this process is very, very sterile, we need to make sure that all of those pathogens are out of that plant. Then we take that explant after we bleach wash it. We move it to something called a laminar flow hood, which is just HEPA filtered air. It's a very clean environment. At that point, we wash it 3 times with sterile water. This is to just deactivate the bleach and get it off of there and stop the plant from processing. At this point, we usually put it on a Petri dish under a microscope, and we excise down to the apical dome. The apical dome is anywhere from 0.1 millimeters to 1 millimeter in size. So to get down this far, we usually have to work with syringe needles, not scalpels, because of how small these plant tissues are. We get down to this part of the plant because this is essentially where the undifferentiated zone is. So undifferentiated just means a mass of cells that they don't know what they want to be when they grow up. So we're going to put them in a lab environment and tell them what we want them to be. Then we take those meristems, we put them on a plate, an agar plate. So agar is a solidifying agent. Think of Jell-O or gelatin. In the agar is where we encase the hormones, the nutrients, and the sugars. And at that point, we have initiated an explant. So after we initiate an explant, it stays in that environment for 2 to 3 weeks. At that point, we do a process called subculturing. Subculturing is essentially just moving from environment to environment. We usually use the same media formulation, so the same hormones, the same sugars, the same fertilizer, until we get the growth that we want to see out of the plant tissue. Once we have this initiation and the plant is good to go, we go through a process called multiplication. So one of the best parts about tissue culture is the multiplying aspect of it. So we can take 12 meristems and we multiply those into 24 meristems. We can take those 24, 48, so forth and so forth. All of this can be done in Petri dishes. So when it comes to size or space restraints, you can fit upwards of 2,000 plants in something the size of a fridge, which really helps with space. So after we go through the multiplication process, we get uniform plants, so all the same size, same health. We take those plants and we actually run them through a process called qPCR. That process tests these plants for anything that could be present, hops latin, fusarium, phytophthora, pythium, just to name a few. We verify that these plants are clean, which means that we successfully did our job. And then we put these plants into their regular environment, which would be called the acclimation period. So whether that be cocoa, rock wool, soil, whatever your preferred medium is, we leave them in there for 2 to 4 weeks, and then you have a regular plant that you can use as you would if it was never tissue cultured before.
How much of the cannabis industry relies on things like this that you do behind the scenes?
Honestly, not a lot, because the cannabis industry is an emergent industry, so it's not really established yet. And a lot of your phenotypes and your genotypes, they use different media formulations. So on a daily basis, I could work with anywhere from 20 to 80 different phenotypes, and each of those phenotypes might need a different media formulation themselves. So a lot of this process is trial and error. I've been doing this for close to 7 years now, and I've run dozens of experiments. We usually start with over 100 different formulations with each phenotype. And of those formulations, we have auxin to cytokinins, as well as different auxins to cytokinins. So you have IAA, IBA. These are things that you see in rooting hormones. Then you have cytokinins, which you don't really see too much of outside of a laboratory environment. There are some products, but they cause rapid cellular division. So we use both of these to dictate what happens.
All right, that was a little bit of a technical explanation here. Now we're going to take a quick break, and when we come back, we're going to talk about the real-world impacts that his work has, be it on medicine or agriculture. We'll be right back. Hello, and we are back. I'm Laurel Stone, joined by Nick, who works in horticulture. Specifically, what he examines is tissue culture for plants. Now, we've talked a bit before about the really technical aspects, and now we're going to look at the real world impact of what his work is. So, Nick, some of the stuff that you do impacts medicine, vaccine production. Tell me a little bit about how your work in a lab with plants translates to the vaccines that we get.
Oh, yeah. So, using tissue culture techniques for vaccine production, usually this involves something along the lines of agrobacterium remediation. So, you have agrobacterium, which is a very unique bacteria. This bacteria actually usually causes tumors within plants, but because of up-to-date research, we've found a way to remove the gene that causes the tumor and insert genes that we specifically want within that plant. Because of that, we can insert certain genes that help with vaccine production. You might have seen this recently with the GLP-1s that they're able to produce on plants specifically. Because of that, you can select the antigen gene that you want specifically. You're going to insert that into the plant cells. Now, at that point, you can reproduce these plants in tissue culture, multiply them out, produce dozens of these plants. Then it's much easier to produce those vaccines on this large scale. Additionally, you have subunit vaccines and you have edible vaccines. These are two different processes. Your subunit vaccines are purified antigen proteins that are produced within the plants. Now, your edible vaccines are more of an experimental type system. They constantly produce vaccine proteins. Now, this would be something that we could put into food production that could then be eaten by a human and would cause those antibodies to form within you naturally. So this would avoid the shot and all the other harmful chemicals that could potentially be within vaccines or shots. And it's just a more organic, natural way that we could develop immune systems to these pathogens. The good thing about utilizing plants for this process and not animals is we reduce the risk of creating a new supervax or superbug or other pathogen that could be detrimental to society. So the advantages of this is it's very low cost compared to animal cells. It's very scalable, and it's the reduced risk of human pathogen contamination and rapid response for emerging diseases. So this is something that we could take and, you could almost produce these things within a couple months compared to years of research that we see. There are some limitations to this process, variable protein yield. So you can't, nature is nature, and we try to mimic it as much as we can through biomimicry, but it's always going to do what it needs to do, which is perfectly fine. That's one thing I love about it. But because of that, proteins could grow rapidly in one plant and not so rapidly in the other plants. There's a lot of regulatory challenges with this, as far as regulations go, where these pathogens are kept, what pathogens we're using. It's a very strict field, as it very well should be. Edible vaccines are not widely practicable yet, so this is something that's very, very new that we're just starting to work on.
What's the growth you're expecting to see in the industry? If you have these new techniques, how important do you think it's going to be going forward?
I think it's going to be absolutely necessary. Kind of touching back, I believe we have about 5% of the Earth's surface is suitable for agriculture. And on a daily basis, we're rapidly growing as a society across the entire world. So we have more needs for vaccines, for one, a rapid need for food production that we can grow on these viable fields. So using tissue culture techniques that would just greatly increase this, especially because we can insert these genes into plants. So we can cause them to be more resistant to salinity. We can cause them to be more drought resistant. On a natural level too, by just increasing them, or in vitro breeding. So in vitro translates to in glass, which everything in tissue culture is in vitro. So we can take these plants, plant A can be resistant to salinity, plant B can be drought resistant. We can breed these internally in vitro and create the seedlings from them, all in vitro. So we can do what would require fields and acres of research in a lab environment and greatly reduce the need for soil.
Now, this prompts me to, a process and expense question. So I'm going to cite cucumbers, that they often require a lot of water to grow. So say we are expecting droughts in the future. We're not. This is an example. I don't want to cause anyone any panic, but we want to make cucumbers more drought tolerant. If you know in your industry that, you forecast that droughts are coming up, again, they're not. This is an example. But how long would it take? Because eventually things, they naturally evolve. But how much would it cost in your lab to make this, to reshape this plant essentially, and then get it out as the regular seeds we as customers buy in the store, not just the farmers? How long would this whole thing take?
From a cost aspect, you could probably look at a couple $100s. This process is not really expensive as you would think it would be. Most of the cost is going to come from, a quality laminar flow hood and your microscopes, as well as your hormones, which honestly are not that expensive. They're really easy to source. There's a couple places I personally like, Plant Cell Technology or Phytotech, both of which the average consumer has access to. They have wonderful reps, and a lot of them have formulations that are already based for your specific crop. So you don't have to do the crazy experiments that a lot of people have to do. Additionally, time-wise, you could look at, it usually takes about 3 months for a apical meristem whole plant regeneration. So if you're really doing just internal breeding, you could probably look at about 2 months. So the good thing also about tissue culture is something called embryo rescue. So it's a process that I like to practice as well, where we have stubborn seeds that they just don't really want to crack for us or open up. So under the flow hood, under the microscope, we actually remove the casing of the seed. We take the embryo out of the seed, and then we plate that on tissue culture, on a plate, and then we grow that embryo out. So we can take these seeds, grow them out in vitro, and then create more seeds in vitro. We can do our plants. We can get up to 48 plants, we'll say. We'll need hundreds, though, for this aspect. But we'll get up to our 48 plants, do the same process, seed out all those plants. Now, all these plants are drought resistant, they're all uniform, and we have our crop ready to go.
So it's easier than one would probably expect.
It is 100% easier than you think. There's a lot of big terms that I've thrown around and a lot of big words associated with it, but down to its core, you're taking a plant organ, you're giving it hormones, which tell it what to do. So touching a little bit on those hormones, we use auxins, cytokinins, and gibberellic acids. All of these are naturally within the plant. So your auxins are mainly produced in your growth point, which is the very top of the plant. Your cytokinins are produced randomly throughout the plant, and your gibberellins are mainly produced in the root system. So when you actually remove the top of a plant or top a plant, dead flower plant, as some of you may say, you're actually removing the auxin growth point. So that's why you're getting branches to form, because now the auxin is removed, which dictates apical dominance. So by understanding these specific ratios, you can tell the plant what to do. A high auxin environment to a low cytokinin environment is going to give you roots, which is why like rooting hormones that you use at home are mainly auxins. Now, vice versa, high cytokinin to low auxin is going to give you shoot proliferation or shoot growth, many stems to grow. If you kind of meet in the middle, you get something that's called callus culture. So callus culture, you see callus a lot of times on plants whenever they're wounded and they kind of build that scar up. It's a callus culture. So those cells are just undifferentiated cells like we talked about earlier, which are cells that don't know what they want to be. So by inducing callus culture in vitro, you can dictate what you want from that callus. You can also store this callus for long-term use in a lab environment, and then just add the hormones or change the hormonal content to that callus and dictate what it becomes.
Now, one of the real-world examples that you gave me of tissue culture is that you saved, well, not specifically you, but tissue culture has saved the banana industry. So tell me a little bit about how that worked.
So one issue with bananas and a lot of farm crops that we use is a process called monoculture, which is using one specific crop in one specific field for a very, very long time. So what happens is you develop pathogens. Fusarium wilt is the one that happened to take out the banana industry for a brief period. You develop these pathogens that are so specialized to this specific crop, you can't really do anything about it. So a lot of people always wonder, like, why does banana flavor not really taste like banana? Well, that's because that banana flavor is based off of the Gros Michel banana, which in the 50s, due to monoculture crop farming, was completely wiped out due to Fusarium wilt. Fusarium wilt is a very, very strong pathogen. It is a huge risk to pretty much the entire agricultural industry. There's a different Fusarium based on whatever crop you're growing, and it is hard to get rid of. It takes around 20 years to remedy any field from this. And then there's no known pesticides, herbicides, to fight it right now. There's nothing registered. So essentially, if you get Fusarium wilt, you absolutely have to use tissue culture processes to get it out. Because of that, Gros Michel being taken out, we ended up getting the Cavendish banana that we know today. And all of the bananas in this industry are now pretty much rapidly tissue cultured because Fusarium is ever present everywhere in the world.
That's really interesting. Again, this is something that not many people know about, but impacts their daily life. Well, thank you so much, Nick. I hope we gave you a little bit of insight into something you haven't heard of before, and I hope you learned something today. This has been Let's Grow There. I'm your host, Laurel Stone, and we'll see you next time.